Okay, so you have now finished your assignment. You know the morphological features of apoptosis. You still know that this is a hugely complex process, but we have a paper available on the website which goes into more detail. Okay, so next we have the connection between caspase 8 and caspase 9. As I said, caspase 9 is activated via the mitochondrial route. Caspase 8 activates the mitochondrial route by the cleavage of bit. Bit gets truncated to T-bit. Truncated bit, T-bit. Bit is a member of the BCL2 family. The BCL2 family consists of 25 different genes in humans and can be roughly divided into anti-apoptotic proteins and pro-apoptotic proteins. Those proteins interact with each other, and it's the balance of BCL2 family members interacting at the mitochondria which determine whether the mitochondria remain intact or whether they will be losing the MMP. So again, like cancer being a disease of disbalance between proliferation and apoptosis, the induction of the mitochondrial pathway of apoptosis is also due to the balance of anti-apoptotic and pro-apoptotic BCL2 family members. Basically, these family members build complexes in the outer membrane of the mitochondria. If you get a disbalance and you have too many pro-apoptotic proteins, those proteins form a pore through which cytochrome C is released, and the mitochondrial membrane potential, the electric currency between the outer and the inner membrane, gets lost. So you get a loss of membrane potential, indicating that you have a breakdown of mitochondrial integrity. And this is normally followed by the release of cytochrome C from the spaces in between the outer and the inner membrane. Now, just because you have activation of caspases still does not necessarily mean that you have cell death. As I said, at every stage of apoptosis can be blocked. And one of the most potent blockers of caspase activity is called X-IAP. It is the X-linked inhibitor of apoptosis protein. And it's X-linked not because it's a member of the X-man, but because it's on the X chromosome. The inhibitor of apoptosis proteins are a family of proteins defined by the Bur domain, and it is actually not quite sure how many of these are inhibitors of apoptosis, despite the name. For example, CIAP 1 and 2 are more involved in RIP-dependent necrosis or necroptosis, while survivin is expressed in a cell cycle dependent manner and is a crucial protein during cell division. Blocking survivin causes mitotic catastrophe. That's also a form of cell death, but is not necessarily considered classical apoptosis. The only protein where I would be 100% certain that it is involved in apoptosis is X-IAP, which can inhibit caspase 3 and 7 and caspase 9. So basically, remembering this slide where I explained to you that caspases get activated via cleavage, you have to now remember that the appearance of cleavage fragments is not sufficient to assume that caspases are active. Those cleavage fragments could be bound to X-IAP and inactivated. So, although caspase cleavage is a good indicator of apoptosis being initiated, it is not sufficient to tell you that apoptosis is processing or proceeding. Okay, good. So now we basically reached a stage where you should be familiar enough with the apoptosis pathway to do our next little assessment, where we ask you to identify key components of the apoptosis pathway.